Okay. Well, let's get started. Are you still in the morning, right? So I should say good morning, right? Are you comfortable? Are you, or is it hot? Too hot? Well, even if it's too hot, I can't help you. I don't control the air condition, so um, I like to move around. I don't know whether you'll be able to. Do you have another mic? That oh, that'll be good though. Do you have one? Oh no, you don't have the one. Yeah, this one. Oh, you control the volume. Okay, good. Can you hear me? Can you hear me? Okay. Um, okay. Can I think you can hear me, right? No? Yes? Okay. Sorry about that. I know we're going to, I'll be talking to you for next 2 weeks, right? So this is the beginning. Make sure we got the right beginning. So how many of you are undergraduate students? I think, okay, raise your hand. So those who are undergraduate, how many microbiology courses you had? You all had microbiology, right? Except this was, you didn't have microbiology? Okay. And assuming rest of you are postgraduate, right? Okay. You didn't say anything, but so when I teach, usually, well, this distance is too far. Are you going to meet all the time in this room or different room? This room? Oh, okay, so they are too far away from me. Um, so I have to speak loud, right? Um, so what I want, if you have any questions, any point, feel free to raise your hand or just yell or scream. Okay? So we want to make it interactive in a sense that makes sure all those things I'm going to talk, you follow, right? Otherwise, if I keep on talking and you don't have any clue what I'm talking, that's not good. It will be just, I will be spending a lot of energy for nothing, right? But I want you to learn something, right? With this course, even though it's going to be kind of like an intensive, right? I mean, usually we take like a 1 semester or 2 semester long to learn all of these materials, but you're going to learn very quickly, right? In a very short period of time. So you need to figure out how to compartmentalize your information, right? And there will be a lot of information, so then you have to figure out what is the most important message in that topic I will talk, right? So the way we will start presenting the materials is that there will be a time I'm going to go like a level, a little bit superficial, means up there, right? Based on your understanding, I'm going to go deep and deep and deep and deeper, right? And so I will go up to a certain level. If you feel uncomfortable, of course, unfortunately, you have to take that challenge, right? And so you got to go and read. And so these slides would be shared with you, right? You're going to get these slides. So you can go over the slides. And when I come back next time, definitely I can take time to address your questions. Will you follow me? Okay, so again, the whole idea being is that this is a special course designed for you to learn about biofilm, right? And the goal would be that make sure you understand, right? Okay, so that we are on clear on that. Um, and other aspect I want to mention is that these lectures, I have prepared about several lectures. You already have the syllabus, right? Correct? So there are many speakers besides me, will be talking to you, and there are information going to be redundant or overlap. But I think that's very good, because in the short period, you don't want to get all the information from me, but there are many expertise, those who are working on specific areas of this biofilm project, and you will gather a lot more detailed information. So don't feel bad that if you heard from me, then other speaker talking about it. Okay? So just think that this is going to reinforce your learning from a different viewpoint, different perspective. Got it? So that's the beauty of this course. I think the way the course has been designed by Professor Goel, and I think I like that, because I'm not the expert. I don't know everything. Okay, I'll just tell you up front. So based on that, we'll get started. So though there are two chapters we are going to talk, introduction and significance. So what I did, that I combined them together. Okay, so whatever I can finish today, I will do that within the time window, and I try not to go over the time, right? And then I will talk about significance as we come along. And like, there will be a lot of repeats also. If I talk some information today, then you will see that I may be talking about that again another 2-3 days later. Okay, so those are the background information. And of course, you are going to have also exam, right? So that will be the last day. Okay, so prepare in a way so that you take the exam and you pass. Okay, so the way I teach my students, I talk to them the day one that I'll give you A on the day one. Are you follow me? So your grade is A. That means that's the highest grade, right? Because all of you are in the academic system. In our system, we call it 4.0. Okay, so I give you A to start with. If you give me a black paper, I'll give you A. The question is, can you maintain that A at the end of the class when you are taking the final? Are you getting it? So I give you A. So all of you have that A grade to start with. Now we'll see if you can maintain that at the end when you're taking the exam. Okay? So that's how I challenge you. So that means you try to keep it. And if you fall behind, that means, of course, that's what you got. All right. So let's talk about the introduction. First of all, I know you're going to listen to me for next 2 weeks, so it's good to know who am I, where I come from. So who am I? You already learned a little bit about the introduction. I don't want to spend any time, but where I come from in terms of U.S. This is the map of U.S. and I'm sure you are familiar with that. And the state called Indiana, and the university is called Purdue University, and it's pretty located close to Chicago. This is Illinois, and it's the Great Lakes right here. So we're kind of like the middle of the state somewhere, but it's pretty north also compared to southern region. I actually, as a graduate student, I started at Wyoming. This is right here, and Wyoming is a wide open space. It's the mountainous area, and this is where I did my college, PhD. And from there, I actually moved to Alabama. Before that, I was in Arkansas, University of Arkansas. Then went to Alabama as a faculty member at Huntsville. That's where the space program. And then, of course, moved north in Indiana. So I've been there ever since for the last 27 years. So total, I've been spending US for 40 years. Okay, so I've been there for a while, but at Purdue University for 27 years. Anyway, so the campus is beautiful. Just to tell you a little bit, these are some of those pictures. This is a huge campus. We have a strong engineering program, agriculture, veterinary medicine, then business, arts and science, and all kinds of programs. So it's a huge campus. These are some of those pictures of the campus. And our logo is called P, which is the Purdue. Purdue, we call it Boilermaker. And this is actually the train it represents. And just a little bit of history about Boilermaker. They used to make actually train engines, okay, and that's why they are called Boilermaker, okay, steam engine. And so we have a car actually representing that Boilermaker. There are some statues in the campus you will see all around. This individual, his name is John Purdue. He gave us the land, and that's how the university was named, called Purdue University. And anybody recognize this individual in the middle? Probably not, but you know he's the first man on the moon, right? Neil Armstrong. He graduated from Purdue University, and so Purdue is a home for many astronauts. They are also currently a very active program, and around the campus, you walk around, there will be lot of statues. So you heard about the book, the Fundamental Food Microbiology. That's the textbook for undergraduate students, those who are learning about food microbiology, foodborne pathogens, spoilage organisms, beneficial organisms, and well described. And there are many undergraduate textbooks, but this is the 6th edition. And you know, Dr. Goel has a copy if you want during this time, if you want to see or borrow, you know, and you are welcome to do that. And then there's a graduate level course called Foodborne Microbial Pathogens. This is completely for graduate level students, means we call it postgraduate. And it's more upper level, describing mechanism of pathogenesis for pathogenic organisms. That's what is being covered in that book. Anyway, these are some of those resources for you to understand. So the way the course is designed, I'm not going to read everything, but you will see that these are the in your syllabus we are going to cover, right? And then including guest speakers, they are going to also cover many part of it. So to start with, what is biofilm, right? And how the biofilm structure look like, and then we are going to talk about also how they help persist organisms in different environments and how they contribute to the disease. Then we'll talk about the mechanistic aspects. Then we'll talk about how do you detect them, assay them, then how do they contribute to the antibiotic resistance. And these are some of those factors in pathogens, right? Then the biofilm also produced by non-pathogenic microbes, okay? Also the beneficial microbes. How do they use that biofilm strategy to help our health, okay? So there are a few things you have to remember. There are good, there are bad, okay, and there are some in between. So that means biofilm, don't take it as a negative way always, okay? Sometimes extremely beneficial, extremely helpful, okay? So some of those aspects will bring that up. And then how do you control or prevent biofilm formation? And then towards the end, we'll talk about the advanced detection methods for biofilm. How do you detect them? What are the current methods? What are the current trends of detecting these biofilms? And in the laboratory modules are designed for you to learn some of those techniques. Okay. Other thing I want to tell you before I start is that there are some of the research. We've been doing a lot of research also on biofilm. I will not say a lot because our lab is designed for working with the pathogens. And so time to time I'm going to introduce some of the research we have done and how that relates to the biofilm, or how biofilm formation helps the disease process, okay? Or biofilm formation can help improve your health, okay? So I'm going to introduce some of our research with the data, okay? So that way you get a more like a broader picture of biofilm, and at the laboratory level or research level, how do they actually work, you know, experimental level, how do they help, or how do they, you know, cause detrimental effects, okay? So this is how it is designed. All right. So let's talk about today's topic. We're going to talk about introduction and significance. So I'm going to talk about importance of biofilms in the food industry, okay? Also in the environment. Then we're going to talk about biofilms in food spoilage. We're going to talk about sustainability aspects. We're also going to talk about the disease outbreaks, and then food safety concerns in biofilm forming zoonotic diseases. So these are some of the topics I'm going to cover today. Okay? Any questions so far? All right. Let's talk about how do we start, right? This is this is a picture of example of biofilm I just started using. Okay, um, this is called a park called Yellowstone. You heard about it? This happened to be in Wyoming. That's where I originally started, and it's a beautiful picture. This is a big park for national park. Okay, and if you go to this park, you can spend like a 2-3 days. Still, you will not be able to completely visit the whole park. Okay, but for people who are working on biofilm, this is the Mecca, right? We call it Mecca, right? Mecca. People, those who are religious, they go to go to Mecca, right? The Islamic culture, right? And so for biofilm researcher, those who are especially environmental biofilm study, this is a perfect example. So this is one of the examples. You can take a look at all those formation of the color, beautiful color, and this water actually hot water. Okay, it's a geothermic activity, and you can see all those colorful organisms they are growing, and the way they sustain their life as it's actually forming the biofilm, right? And Dr. Shukla is going to talk about a little bit also environmental side of biofilm. So this is a classic example, the biofilm, right? Let's look at another one. So this is continuous geothermic activity, the water coming out of the ground, and it's like a volcanic activity, but it's the water, hot water keep coming, and you can see this formation, this structure actually contributed by microbes. These are all microorganisms, forming a nice tight structure, and then of course they mineralize and they calcify, and eventually this type of solid structure is hard to break it. It's not fragile. It's extremely solid structure. So these are examples of how the environment or microbiology or biofilm look like. This is also Yellowstone, and this is also another one. You can see this. This is called bacterial river. Okay, if you look at the bacterial river, it's very colorful, right? And you can see this is formed by algae, the bacteria. They're all collectively growing together and forming a river. And so that's also a prime example of biofilm in nature. Okay, and this is again in Yellowstone. And if you go to that park, obviously you will be just amazed how the microbial activity form this natural structure. It's just breathtaking. It's beautiful. You can spend hours and hours watching these, and you'll be amazed that how nature take these microbes or microorganisms under these conditions, and they're growing and they're surviving, and then forming this beautiful biofilm-like structure. Okay, so this is an example. How about this? This is a pipe, right? Water supply, and you can see this built up. This is also part of the microbial activity. And they are also forming biofilm or biofilm-like structure. So, best form of definition of biofilm. One of the things I learned from my professors that microorganisms never exist in isolation. Like we as a single entity can be present, right? But microorganisms in nature, they never a single cell. They're always biofilm former. They are with a community with other microorganisms. So always microbes exist as biofilm. So that's not a surprise. We are learning it. If you look at the history of the biofilm, I mean, you can go back to Leeuwenhoek or Pasteur. They knew the microorganism exists on surface. They are actually forming certain biofilm. They may not have coined the terminology as a biofilm, but it exists for centuries. Okay, and in environmental microbiology, obviously, people started to understand that microorganisms present on the environment, they are in the form of biofilm. Okay. In the medical science, medical microbiology, clinical applications, the first, I think the report was 1985, they reported that the biofilm actually responsible for causing many diseases. And the first disease they studied, that is a lung disease called cystic fibrosis. They found Pseudomonas is the main organism responsible for that disease. So that's when the medical biofilm terminology was established in 1985. Okay, and ever since then, all those other field of areas, okay, environmental, starting with soil, and then you can say plant, then also in the food sector. I think I would say much early, late 90s and early 2000s, people started to study biofilm and their implication in diseases and also foodborne, contamination, and so food processing plants. All of those factors begin to be reported during that early 2000s. So the take-home message is that the biofilm is a natural process for microorganisms, and they form called polymicrobial. Okay, the terminology you might find it called polymicrobial. And so this is an example. So this is the aggregation of cells, and often many species into a complex structure on solid surface. So again, this one I would say a little bit biased. They say called solid surface. Okay, yeah, the biofilm formation cannot happen in liquid. Okay, but sometimes they can break away, and under certain conditions they may form it. But the solid surface is pretty much a kind of like a standard practice that microorganisms get together, they form a community of organisms, and solid surface is the primary requirement, right? And so here you can see this. One of the aspects of this picture, I liked it, is that if you look at these organisms, different organisms, and then can you see this group? It's like a channel, okay? And this is beautiful. I think as you talk more and more, you'll find out the biofilms are designed in a way that these organisms can survive, they can protect, and also they can transport nutrients through the system. It's called channel, right? And so it's kind of like active whole community. They are sitting in an environment. It's like, think about it. If you establish a village, right? Think about it. First establishing a village or community, then you need to have a roads, right? To go. You need to have a water source. You need to have a food source. All of those components needs to be in that environment. If you use that analogy, think microorganisms also need that environment, right? They need to be in a community. They need to make that community. That make sure the community has access to water, the food, right? And they can, transport materials across the whole community so that it benefits the whole community. Getting it? And so microorganisms, that's why they make this type of biofilm. And now we are beginning to understand what really goes on inside the biofilm. Okay, this is also another example. You'll see some of those, scanning electron microscopy pictures. These are also, all varieties of organisms. This is also another two different types of organisms forming biofilm. Okay, so whenever we talk about biofilms, what is the benefit, right? So it's a community of microbes held together by certain matrix, right? We call it extracellular matrix or ECM. This is also called EPS, extracellular polymeric substance, EPS. And usually that EPS polymeric substance is consists of what? Polysaccharides, you need carbohydrate which is very sticky, right? Then you need protein. Protein also part of that. What else? We have also lipids, fat or lipid molecules, and then also eDNA. eDNA, you will see that also, we'll talk more about it. It's called extracellular DNA. Okay, the DNA is one of the components of the cells, right? We all have DNA, but when they make this biofilm, the eDNA play a very important role, and it's become very sticky, extracellular DNA. And so some cells in the community, they die, they release this eDNA, and eDNA become part of the community structure. Okay, again, we'll talk more about these details later on. It protects from sanitizers. So they form biofilm. If you are trying to kill them, sanitizers, chemicals, drugs, antibiotics, they cannot penetrate. maybe they will kill the organisms which are on the surface, but not necessarily the organisms which are deep into the community. So the protection, you need that protection from having the biofilm structure. Other things, shelter for pathogens. Think about it. So the biofilm, as I said, is a polymicrobial, right? Different types of organisms can form biofilm. But sometimes this polymicrobial biofilm, not everyone is bad candidate in that community. Some are bad, some are good. Some are there for certain purposes. But this aspect of it, you have to remember that. So whenever the biofilm is formed, sometimes you could find that pathogens like to be in that biofilm. You see this? And that aspect you need to pay attention to. You may isolate a biofilm from a hospital setting or from a patient. You might find, oh, this is organism may not be the one causing disease, but have you looked closely? It might have some pathogens inside that community. So it's a good place for pathogens to be in. So it's a good shelter. Nutrient source. As I said, biofilm themselves can serve as a food. Okay? Because they are making these different components. You know what microbes eat, right? Everyone knows what they eat? We know what we eat, right? What do you have to give them to survive? Right? First thing would be carbohydrate, carb, right? They like carb. Okay, simple sugar would be the best choice, right? Glucose, sucrose, fructose, whatever, and they like that, right? So the nutrients, right? And so, as I mentioned, some of those things on the top, the community, they're making these molecules, and a lot of these molecules serve as a food. Okay, as they help them together, but they sometimes also they help them to survive because that's their nutrient source. Okay, what else? Persistence. Okay, biofilm, of course, they survive for a long time, right? It's very difficult to get rid of, and it helps the microorganism to persist on that environment. Okay, this terminology persister, we'll talk a little bit more. I think some of you graduate students will need to understand more than the undergraduate, but I will try to explain you molecularly what happens. So persister is a phenotype, is that these organisms, if you try to kill them with antibiotics, they are not antibiotic resistant, but they can survive. You get it? Something I said. They are not antibiotic resistant, but they can survive antibiotics. Okay? A certain population of cells, when you try to kill them with antibiotics, maybe all of them are sensitive to antibiotics, right? They are all going to die. But certain population within that community, they are not going to die to that antibiotic. But they convert into called persister cells. They can persist in that environment. Have you noticed that when you go to a doctor for chronic diseases, and they will give you antibiotics, right? But then you just never got cured. You always have that persistent infection. Have you noticed that? Or sometimes if you talk to, there are some diseases, this is very common, okay? So there is a microbacterial infection or some of these other ones, even though those organisms are not antibiotic resistant, but they become called persister cell. And this is a new phenotype or phenomenon been described last 15, 20 years, and there's a lot of studies going on that. But this is an extremely important. And these persister cells, if you try to kill them immediately, they are not going to respond. You cannot remove them. So they are part of this community, that biofilm community. Everybody clear? Again, we'll spend more time on those. Other aspect, we said that biofilms are resistant to antibiotics. Okay, so you are trying to kill them, and they actually can absorb antibiotics. If they absorb antibiotic, the biofilm, it doesn't reach to the organism. Okay, what happens? What else can happen in the biofilm? What about their status? Do you think they are active as if they are outside in a healthy environment? What do you think? Usually they are not that active, metabolically active. What does it mean, metabolically active? If I say this terminology, what does it mean to you? Someone said something, but I couldn't hear you. So metabolically active means what? They can uptake sugar, right? They can convert it into byproducts, right? Whether it's acid or gas or whatever may be, right? Or alcohol or whatever. Right? That's these cells are active, right? So that means they can uptake nutrients. Everybody clear? Metabolically active and convert it into byproduct. If I say metabolically poorly active or inactive, but they are not dead. Are you getting it? So think about the hibernation. You heard about the hibernation? The animals go into hibernation, say during winter months, right? So they kind of control their metabolic activity. So they don't need the energy. They don't need to get out, right? Snakes do that. A lot of wildlife animals do that, right? They can stay alive, but not metabolically or active. They are not doing anything, right? So with a low amount of food, they can survive extended period of time. So if you think that hibernation, the microorganisms essentially they hibernate in that community. So if I want to talk about the antibiotic resistance, what do you need to have in order to kill this organism? Then antibiotic needs to get in, right? But if you are not taking in anything, are you going to be susceptible to antibiotics? Everybody see this picture? So this metabolic activity is very important, okay? And I think that was coming. So I started talking about it because I wanted to ask you, it's low metabolic activity. That means they take less antibiotics. Okay, therefore, you cannot kill them. It's very difficult. Okay. And also they play important role in pathogenesis. So you talk about they will adhere, attach, and then also they can evade immune system. Okay. So these are some of those attributes. You can see this benefit to the microbe. Correct? All right. So since speaking of antibiotic resistance, which is going to be extremely important, we will be talking about, there is a separate chapter on this. As just to give you an introduction on the antibiotic microbial resistance. So you can see all of those outline I provided. They are talking about antimicrobial resistance. So presence of persister cells we already talked about, right? So persister cells, they are not responding to antibiotics in population. High cell density and quorum sensing. We'll talk about that more in detail. Quorum sensing. You heard about quorum sensing, right? How many of you heard about it? Quorum sensing. Raise your hand. Okay. So few of you. So quorum sensing. Let's talk about that. How do you talk to each other? Do you talk? You're going to talk, right? What else do you do? Nowadays you text, right? Right? Even if you are sitting next to each other, you text, right? So that's a communication, right? So the quorum sensing here is a microbial communication. They talk to each other. And they don't have cell phones, so they can't use they cannot communicate. So what they will do, they will release some small molecule. Okay? As soon as that molecule is released, then you say, oh, he's alive, he's sending me some signal. Okay? And then that microorganism is going to respond to you. Okay? So what is quorum means to you? Quorum, when you say quorum, what it means? Have you done any meeting anywhere, anytime? Like you have a group, club, any of those activities, right? Sometimes you have different types of club, right? I mean, if you say you're athletic club, right? So you have certain members, right? And so what do you do in a club? Say, okay, I need to make a policy decision, right? So okay, from tomorrow or from next month, we are going to meet at this time, right? And so okay, this is approved by the committee. Are you seeing what I'm talking? I'm talking about totally different things. I'm not talking about microbiology. It's a human, leadership interaction. So if you have a faculty meeting or professors meeting or student meeting, right? So we need a certain number of people, right? And that number of people are going to vote on that. And then if you have a certain number of people, they vote and then they will pass that. Then it becomes a policy. So in order to pass that, you need to have a certain minimum number of people, right? And that's called quorum. So when you are creating a meeting or conducting a meeting, you want to have a quorum, means a minimum number of voting members, right? And so here, that same information that we started using in microbiology. We call it quorum sensing. Quorum sensing means the microorganism trying to figure out what is their population in that community. Why do you need to know? Why microbes want to know why do we have this many number of microbes needs to be there in the community? What is the single most reason? Say that loud. Resource, right? What is that resource? Nutrient, right? I would start there. Food, right? We need food. And so in order for microbes to grow, they need food, right? So if I don't have enough food, do we need to grow or multiply? No. Right? So that's the information. How do I communicate to that other member who is about to divide or reproduce, right? Are you getting it? If you have a limited resource, you don't want many of them to grow, multiply, right? You want to limit their growth. So stop dividing. We don't need you anymore because I don't have food to feed everybody. Okay? That communication is very important. So the cell density, cell mass. Are you getting it? If you have a 5 microgram glucose in a test tube, you add some microorganism bacteria, they started growing, right? And then after some time they're going to stop because the nutrient is going to exhaust. So if they are in a community on a biofilm, if that same scenario happens, you need to talk to the friend, right? Stop multiplying, right? Stop reproducing. That way we all can survive, otherwise we all going to die. That's not the goal, right? We want to survive. And that's when the quorum sensing aspect is very important. So that means the microbes realize that we have a limited resource. That's one of the things. It's a stop dividing. And how do you tell them to stop dividing? Well, we've got limited number of food, so they will produce some small molecule. And that molecule is going to bind and tell the other person, okay, I got this message. You already told me there is less amount of food. I'm not going to multiply. I stop. Got it? That's how we are all going to survive. And that molecule is the communication. You see this? The microbes talk to each other. That's called quorum sensing. So 5 of you raised hand. Now how many of you are going to raise hand now? All of you? Great. So that's the whole purpose. Are you still not clear? We'll talk about that again. Okay? It's the quorum sensing. We understand that environment and we need to act accordingly. And the microbes, we thought they are just little microscopic organisms. They are just dumb fellows, but they are not. They are pretty smart. They are extremely smart. Okay? And that's why quorum sensing is very important. So I know I spent a little time there. Hopefully by the time we talk more about it, you will learn a little bit more. Oxygen concentration gradient, right? Metabolic activity, the mechanical aspect of it, right? A lot of these factors can help microbes to become resistant to antibiotics. Because you know that antibiotic resistance is a huge problem, right? We all recognize that in microbes or microbiology, it is an extremely important. So this actually summarizes some of those. And then again, like I said, we're going to talk same thing few times. Okay? If you didn't follow it today, hopefully 2 days later or 3 days later or next week when you talk about antibiotic resistance, you'll get a lot of ideas. Got it? Okay. Not to dwell on too much. So the antibiotic resistance problem, AMR call it, antimicrobial resistance. This is the largest global threat. Predicted mortality rates of about 10 million by 2050. So many people are going to die because of the antibiotic resistance. Okay? So biofilm is one of the major contributors to the resistance. Got it? It's not just the genetic manipulation of the gene, which everybody try to study that. Many people study that, right? But this is the other factor. If they are metabolically inactive, they don't take up the antibiotics, they are not going to die. See this? There are many factors you need to understand. And so it's the compilation or combination of a lot of factors. Multi-drug resistant superbugs, Salmonella, Staphylococcus, extended factor we call ESBLs, that group. These are Acinetobacter, Klebsiella, Mycobacterium, Neisseria. These are all these problems. And then, of course, WHO report that about 490,000 cases of multi-drug resistant TB. That was in 2016 report. So that means it's a huge issue. Again, you need to understand from the, since we'll be spending a lot of time on biofilm, think about how biofilm contribute to that resistance. Okay. All right. So microbes. Then, when we talk about microbials, biofilms are the major sources of pathogens in food. That's true. We'll talk about that. So let's go to ABC of microbiology. Okay. I'm going to spend time a little bit where those organisms come from, how is the life, right? So we know that there are three domains of life, right? Bacteria, archaea, eukarya. I'm sure all of you knew that, right? Three domains of life. So the bacteria, archaea, they are major biofilm formers. Also the fungi, which is part of the eukaryote, they are also biofilm formers. And then the parasites, we'll talk about that. So when you talk about biofilm, we are not just going to focus on bacteria. We're going to talk about also fungi, parasites. They are also part of the biofilm forming community. They all also contribute to their individual niche in the host or in the food or in the environment. Okay. So bacterial biofilms, we'll talk about that more, we'll learn about more, right? And if you look at these different types of pathogens come through food, and these are a huge list, right? I'm sure you will be able to recognize some of those organisms. Okay. These are we consider called foodborne pathogens. What we learn about them, that any type of food we eat, I'm talking about human or animal feed, and a lot of these pathogens come through. Okay. And just because my interest is in the foodborne pathogens, I'm going to highlight some of them. Aeromonas. Sorry, water got into my. So Aeromonas hydrophila is a waterborne pathogen. Bacillus anthracis, called anthrax, is also animal called zoonotic pathogen. And Brucella, Campylobacter, Clostridium, Chronobacter, Escherichia coli, Listeria monocytogenes, Mycobacterium paratuberculosis, Salmonella, Shigella, Staphylococcus, Vibrio, and Yersinia. These are some of the common foodborne bacterial pathogens. So when you talk about these foodborne pathogens, let me give you just one example. Again, I will study more. You recognize what is in the left? That's called cantaloupe, right? We have cantaloupe in India. You have like a melon family, right? You know watermelon, right? All those other melon family. We have, yeah. This is cantaloupe. It's very popular. It's the same family called cucurbit family of fruits, right? And you look at the structure outside. If you follow the electron microscopy structure, and it has followed a net-like structure outside, and it grows like watermelon, right? Any melon or pumpkin or squash grow outdoor, right? And we are having a lot of problem with this. Okay, I mean US. We are talking about, we had outbreak with, of salmonella with the cantaloupe. We have also huge outbreak with the listeria from cantaloupe. A lot of people died actually just eating cantaloupe like this, and listeria outbreak. So look at this, how this beautiful sweet fruit become a major concern for health, right? People died for eating cantaloupe. So if you look at that, the structure, you can see this net-like structure, and then there's a valley. So this is the part of the network, and this valley, if you look closely, you can see this structure, the plant-based structure. And what it looks like, it's a pretty rough structure, right? And is that a major source of pathogen in the cantaloupe? Well, if it's outside, right? So let's look at it. There are some of this work from our collaborators at Purdue University. So he studied how listeria actually colonize or form biofilm on the cantaloupe. Okay, and so this is just 2 hours of exposure. You can see a lot of listeria bound. Just 2 hours. So that means the environment is perfect for listeria to colonize. Okay, essentially we're looking at the forming biofilm on the structure. And if you look at it 12 hours, you can see more. And these are the 48 hours. I think you can see clearly this is a huge layer of bacteria sitting there, right? Then again, we don't eat the skin, do we? No. So how do Listeria get into the cantula? So we have bacteria. The outbreak the way happened, and there's a lot of research happened. So everybody is trying to figure out why this cantaloupe become a major source of Listeria nowadays. Of course, Listeria, just to let you know, these are environmental organisms. You'll find them in the soil or plant material. This comes from animals, and these organisms are already there. So if the cantaloupe is growing, if the conditions right, remember I mentioned a little bit about climate change, temperature rise, all of those factors play an important role. More and more outbreak we are going to see. There's no end to it right now, the way everything is going. So you guys have to prepare well to prevent from getting killed or die, right? A lot of people die from this type of just eating cantaloupe. So you don't want to be killed, right? So let's use your common sense. The organisms outside, nobody eats the skin, but how do these bacteria get inside, right? Usually we try to clean it, wash it, use chlorine water, ozonated water, a lot of those disinfectants we use to clean the surface, but a lot of times they could be still there, right? So how do you make a this is how we cut it? You use a knife. What are you doing this by doing this process? It's extremely common sense. Tell me what is happening. You use a knife. So what you are doing actually, if you have a biofilm sitting outside, right, and using knife, so knife is helping the microbes to eat. You see, that's very simple practice. You don't think about it. Any fruit, I'm giving this as an example. Fruit, you cut, the microorganisms are already outside, right? And when you cut it, these organisms are going to get in. And what happens? If you cut inside, you already know the inside is very juicy, right? It's rich in sugar. Who likes sugar? Microbes, besides us. They like sugar. Anytime, anywhere you give some sugar, they are going to grow, right? That's their and they are going to grow. And so when you cut it and living there on the room temperature for a few hours like this type of weather, oh, they are going to just multiply so rapidly. Okay, so most of the problems we have when you buy this cut fruit, okay, and you need to take a lot of precautions to prevent it. So can you see this, how the biofilm contributing to people's death or suffering? You see this one? So this is a direct example. And so they are trying to find a different way to remove this biofilm. All those different strategies. We'll talk about some of them, okay? Use some food sample, and then how do you remove this biofilm? That way you can prevent it. What about yeast and mold? We talked about bacteria, yeast and mold, they also form biofilm, right? Look at this. These are Aspergillus, Candida, Coccidioides, Cryptococcus, Mucor, Penicillium saccharomyces is the yeast. So these also can form biofilm. And this is a classic. We had a tomato in our basement. We left it for a while. I forgot to, bring it and eat it. And then I went there and downstairs. Usually I don't check it. And I went and picked up this tomato. Look at this. What happened? All the mold growing, right? Mold is a good spoilage organism. And if you think about, that's a beautiful biofilm, right? They form a nice structure. So if you look at the yeast and mold, these are the common structure of the yeast, the mold. And you have seen this, the mold species, Aspergillus, Penicillium, and they make mycotoxin, okay? And mycotoxin is a deadly toxin. It causes cancer. Okay, a lot of times you eat certain food, and if you continue to consume mycotoxin, there are chances that you are going to get cancer, especially the Aspergillus toxin or aflatoxin, which is a type 1 carcinogenic agent. How many of you thinking that eating food can give me cancer? We don't link that, right? Think about if you continue to eat mycotoxin tainted food, like a peanut, a lot of those nuts, our favorite place for a lot of mycotoxins. Okay, so there is a certain standard. You try to monitor it before you consume it. So this is an example. They can grow, they can produce toxin, and that mycotoxin you cannot heat inactivate it. You can cook it many times, you can still cannot inactivate mycotoxin. Okay, there are special way you can inactivate it, but very rare. But if consuming mycotoxin built up in your system, it can cause you skin cancer, mostly the liver cancer, hepatocarcinoma, also the kidney cancer. Okay, and all of those starts with the mold growing, forming biofilm, and producing toxin. This is the picture of a yeast on the left, and they also form biofilm. Okay, biofilm produced by Saccharomyces on the left, and the Candida albicans actually it causes disease, serious disease. Okay, it's a mold-induced disease, and they are also notorious biofilm formers, big biofilm formers. We'll talk about some of the clinical disease. Then we already talked about that one, the biofilm. The parasites. Okay, think about parasites. We ignore that parasitic diseases, but not in India. I know we have a lot of parasitic diseases because of the higher temperature, right? And the tropical countries, they suffer from a lot of parasitic diseases. Okay, so we are not talking about metazoan parasites. We are talking about unicellular parasites, means single cell parasite, and those are like Cryptococcus, Cryptosporidium, Cyclospora, then Entamoeba, Giardia, Isospora, Toxoplasma, Leishmania. These are single cell or unicellular parasites. Okay, and they also form biofilms, and they become a big part of the problem. So there are many diseases. We'll talk about that. How are we doing with the time? Probably he's not keeping track of. 12:30. We'll finish at 12:30 or what? What time are we supposed to finish? 12:30. That's okay. I don't want to because we want to stay on schedule. So let me finish the first part of the introduction. This is an example of the unicellular parasites. So we're looking at Giardia, Entamoeba, and this is Toxoplasma. These are the unicellular organisms. Okay, and they form these parasites, the biofilm. We'll talk about some of those as we go along, the diseases caused by these type of organisms. You heard about tapeworm, right? Tapeworm is not unicellular. This is metazoan, large parasites, and people suffer from, especially unhygienic conditions. Tapeworm is kind of dangerous. Then also roundworm, those are the long one, but they don't form biofilm. These are like large ones. But I'm talking mostly about the unicellular on the top. Okay, so how actually the biofilm help the parasites? Okay, unicellular parasites. So if you look at there are three functions I listed. So parasites, and they can sometimes destroy the biofilm, okay, because they make enzyme. They produce gastric the enzymes, and that enzyme actually can destroy biofilm if the biofilm is produced by bacteria. That's number one function. Number two, parasites influence biofilm formation. So some of the products they make that can help biofilm formation. And then the last one, biofilm can protect parasites. So if there is a biofilm formed by bacteria, and then parasites can be hidden. Remember I was mentioning before that it's a shelter for many pathogens. They can be all in one environment. And so parasites, and they can definitely survive inside this bacterial biofilm, and then eventually that can play an important role in causing disease. Okay? I want to point out that every slide, most of the slides has certain references, and these are all recent references. If you are interested in learning more, always you can go to that article and find more information. Okay? Keep that in mind. Okay, so this slide, I think this is talking about fungal biofilm. Okay, and what they are looking at is that there is a huge application of biofilm in the industry. And so if you look at that, biofilm of fungi helps citrus resist acid rot. This is kind of interesting. That's the reason I put it up there. We talked about parasite, but this one mostly for the fungi. I should have switched the slide before biofilm was attached to the water pipe. So they created water pipe. That happens. Biofilm accelerates the corrosion of food processing equipment. So the application, in a sense, both negative and positive. Okay, it's saying that, well, if you have a biofilm, we know that that's going to corrode equipment. Okay, because of the way they behave, they eat up the equipment, right? And like I showed you the pipe, biofilm contamination of implanted medical device. That's of course a huge problem. We'll talk about that. Biofilm production generates secondary metabolites. Definitely that does. Biofilms are used to feed broiler chicken. They use biofilm to feed the animal. Biofilm are used to treat sewage. Biofilm are used for biological as an alternative to pest and disease drugs. So huge applications you will see with the different types of biofilm. Let me see. Maybe this is a good place to stop. Then we'll talk about biofilm. One of the things I want to send you the message, like saying that not always bad. Okay, so biofilms, if you look at the beneficial, spoilage, and pathogenic. So to me, I think the beneficial biofilms are helpful. We need biofilm to be produced, can help certain diseases, not diseases. We should not talk about disease. Certain fermentation process, okay, certain gut health or improve health. You need biofilm formation characteristics. Spoilage also part of that. I showed you some picture. They can spoil food. That affects the sustainability aspect. If the food spoiled by biofilm formation, and then of course the pathogenic activity. Okay. So we've got some slides. We'll talk about that. And then I know today is the first day. Probably everything time-wise, but hopefully from tomorrow we'll be on time, on schedule. And then again, make sure when you go home, or home means your residence where you are staying, make sure go over this slide. You have a good familiarity with these slides. That way, next day when I come and talk about it, so that you have a good idea what I'm talking. Okay, and I need that feedback. Otherwise, your learning won't be heightened. We need that you go through these slides, even if you don't understand any of those slides, that tomorrow's lecture, but just go over that. Okay, it gives you familiarity, and that way when we talk, you will have a better grasp of material. Got it? So I think I'm going to stop here, and then I think you have a lunch break, right? Okay. And then after that, Dr. Shukla going to talk. Okay. Any questions before I go? You should have a lot of questions. Again, prepare, bring questions. I want to see your learning. Because if this is more my way of teaching is that I don't want to keep on talking, and then you will be sitting there listening and not really understanding, or you don't want to ask questions. Don't feel bad about questions. Okay, I never get offended by any question. There is no question called stupid question. You knew that? You heard about it? Stupid question. People are asking stupid questions, and it may be a stupid question. I shouldn't ask it, right? And that prevents you from asking the question. But there is nothing like that. There is no question is called stupid question. You heard about it? So questions are always good. Any questions? No. That's why you have to ask the question now. You ask me now. I will ask you later, right? Or we are going to ask you later. Go ahead. Oh, okay. We'll talk about that a little bit more. eDNA, just to simply, what I was trying to mention, that DNA molecule is very sticky. Okay? If you break open a cell, the DNA gets out. They are extremely sticky. And so when this in the community, some cells die, some remain alive, and their DNA gets out. Okay? And that eDNA, which is called extracellular DNA. It's kind of funny, right? E stands for not electronic, it's extracellular. Everything nowadays E, right? So this is extracellular DNA comes out, and it makes it sticky, so it helps the biofilm to stay together. And that's what it is. And the DNA molecule, in general, they are very sticky. Okay? The type of skin infection we get, which organism cause most of the skin infection? Anybody said Staph aureus? We're going to talk more. carbuncle, boil, right? Lot of the skin is the toughest place, right? I usually use this example. Staph aureus makes a lot of nuclease enzyme, tons of. The reason they do it, one of the things is that as soon as they make this boil or carbuncle, you have the skin infection, right? Usually come from staph, and you squeeze it, the white pus come out. You know what I'm talking about, right? We all suffer from. This is nothing new. If you are a little bit not taking shower frequently and all kinds of stuff, your skin, all are inhabited by staph aureus, and skin infection is very common. I know the mold, they also can cause it. So the staph aureus, the reason I brought this up, they make a lot of nuclease. And the reason they make a lot of nuclease, because as soon as the infection happens and this DNA gets out, that can impede the progression of the disease. So that's why they need to make the nuclease to liquefy the DNA. So when you squeeze it, you will see all of those DNA are no longer fresh DNA. It's a lot of dead immune cells, a lot of dead cells, and then already bacteria can infect much easily. Because if you break open one cell, the DNA is nasty. That I know I gave you a long answer, but the whole idea being is that this is a very sticky molecule that helps the DNA, the biofilm formation. Okay. Any other questions? Yeah, go ahead. I think you have to speak louder. How benefit or for treatment? For treatment? Sewage treatment. Oh, I see. That's a very good point, though. Just simply, I can tell you that the biofilm being there and producing enzymes. That's a good point, though. I don't know. I have the answer. I'll find out that answer. The sewage application. I think it will be like a lot of enzyme production, and enzyme production may help clarify certain molecules. This is the environmental side. A little bit less familiar. You may have that answer. But I think I'm thinking more like a fermentation process I will talk about, and there is a biofilm formation, help fermentation, for example. But I'm thinking more like a byproduct production. The organisms being there, producing certain enzyme, are they clarifying certain molecules from the sewage, removing certain undesirable molecules. Okay, if you don't have any other questions, then we'll go for the next page. All right, I'll see you. I think I'm done for today, but then we'll start tomorrow morning.